This is lecture 27, and we're going to be exploring the motivated behavior of hunger and those internal and external factors that are involved in our eating behavior. And along the way, we'll also talk about a really interesting eating disorder because I think that it'll help us to understand some of the controlling mechanisms that are involved in this behavior. And so we have a lot that we want to accomplish in this lecture. So let's begin our analysis by taking a look at this little model that you see here that I think helps us to understand a little bit before we get into some of the nuts and bolts of studying hunger. Hunger is considered to be what we would call the prototypical motivated behavior. And what we mean by that is that, there's very well-defined external and internal factors that control our eating behavior. And they can be seen in this model that you see here. You start to get hungry, you start to get those feelings of hunger with oftentimes a growling stomach and a feeling of weakness. And as you grow hungry, you begin to recognize that you need to eat. You need to begin to seek out food and start to eat. And then once you do eat, you eventually become satiated, and there are signals, very important internal signals that are involved in that satiation. You become full. And now you are between meals. Again, satiety is the satisfaction that you feel between meals. So, and then this whole process, this whole cycle starts in again. So we need to know what the various factors are that contribute to this, the external factors and internal factors that are responsible for controlling our eating behavior. I want to talk about a pioneering psychologist and his work in this area. His name was Stanley Schacter. Very distinguished psychologist for many years at Columbia University in New York City. And he really made contributions in many different areas. His studies of birth order, of the factors involved in smoking and emotion and addiction are very well known. But I think that his work in the area of obesity is probably some of his best work. Because of the very inventive experiments that he employed in this work. He really was the first one to identify that obese individuals seem to be very different in terms of the factors that control their eating behavior. They seem to be very, very sensitive to external stimuli, whereas normal individuals respond more to internal biological cues. Now, that's an important finding because that finding in and of itself may help us a great deal, obviously, in terms of trying to control abnormal eating behavior that leads to obesity, but also gives us some important clues as to the stimuli that are involved in normal eating. I want to talk about his very important experiment on time perception, in which he was evaluating this time perception in both obese and in normal subjects. And his hypothesis was this. He thought, well, normal subjects, he thought, are eating when their bodies tell them to, when they're feeling the weakness in their stomach, for example, that then in turn is stimulating neural pathways that go from the spinal cord to the brain. And that's what normal subjects respond to. They eat when their bodies tell them to. So again, they're responding to internal biological controls. On the other hand, he says obese subjects, I think, eat instead by the clock. They really don't depend so much upon their stomachs and their feeding, neural feeding circuitry. And he set out to try to examine this hypothesis with a really interesting experiment. This experiment is one that is described as a basic 2x2 experiment. Here's how the experiment went. Subjects were asked to participate in filling out a very lengthy and boring questionnaire in a room in which they could not tell time of day. There were no windows in the room, there were no clocks in the room, there was any kind of device that would help them to tell time, like a watch, for example. This was removed prior to them going into the room to fill out this questionnaire. Then, after the subjects were involved in filling out this questionnaire, they were led into another room, and this room had a series of tables that had bowls of good tasting crackers. Now, some of the subjects in the experiment were normal subjects based upon their age and their body size, and other subjects were obese subjects. That is to say, they were more than 2 standard deviations above the norm in terms of their body weight. So we also are going to tell some of these subjects in each group that it's before their normal dinner hour, and other subjects we're going to tell that it's after their normal dinner hour. So again, this is called a 2x2 experiment. Again, normal subjects and obese subjects, and the subjects being told, half of them, that it's before their normal dinner hour, and the other half being told that it's after their normal dinner hour. And they look to see how many crackers the subjects eat. And normal subjects, it didn't matter if they were told that it was before their dinner hour or if they were told it's after their dinner hour. They ate an average number of crackers. Obese subjects, on the other hand, when given information that it was before their normal dinner hour, they ate very few crackers. But if they were told that it was after their normal dinner hour, they ate a lot of crackers. So again, this prompted Schacter to think that, there seems to be, these 2 different mechanisms that ordinarily are involved in our eating behavior, but the one that predominates in the case of an obese subject are these stimuli that are coming to the individual in terms of time of day. And you can see this very dramatic effect here in obese subjects of being told that it was before their normal dinner hour, in contrast to after their normal dinner hour. So indeed, a very interesting experiment. Schacter also looks at some other characteristics of obese and normal subjects. Obese subjects tend to be plate cleaners. Normal subjects, on the other hand, are much more likely to leave something on their plate. Obese subjects are affected by portion size. If they are given a large portion, they eat it, but they seldom ask for more. If they're given a small portion, they'll eat it, but again, won't ask for more, whereas normal subjects are much more likely to ask for more. Obese subjects are affected by the taste of food. If it's really good tasting food, then they eat a lot more of it than do normal subjects. If the food is bad tasting, and one way in which they manipulated this was to lace some of the food with quinine, for example, which is very bitter, obese subjects ate far less of the food than did normal subjects. What Schacter also found in these experiments is that obese subjects were much less likely to perform any kind of physical labor in order to obtain food, or if they had to suffer mild amounts of pain in order to get food, they simply wouldn't do it. So again, we have this picture that is emerging of obese subjects really responding much more to these external kinds of factors as opposed to any kind of internal biological cues. So those experiments become very interesting for some of the work that I'm going to tell you about that has looked at animal models of this. So we'll return to this and these findings a little bit later on. So what are the signals for hunger? Well, certainly there are many who believe that the growling empty stomach is important, but if you cut the nerves that go from the stomach to the spinal cord to the brain, individuals still report being hungry and regulate their food intake just fine. External factors, the smell of food, certainly the taste of food. You know, these certainly influence our eating behavior, but subjects, and you know, some of this work is with animals, some of it is with humans. If they can't smell or they can't taste, they still regulate their food intake just fine. And certainly there are cultural factors, you know, habits, customs. You know, in our own country here in the United States, you know, we're used to eating 3 meals a day, with the large meal being in the evening. In other countries, namely European countries, their large meal is at noontime and a much smaller meal in the evening. In certain Asian countries, for example, they eat many small meals over the course of the day. So again, habits and customs certainly, you know, also play a role. So again, we talk about what factors seem to be necessary, which ones seem to be essential in terms of our eating behavior. That leads us into an examination of a very interesting eating disorder, which is called anorexia. And without question, thin is in the world today. You only have to look at the models and how thin they are, that you know, major companies that are advertising their clothing, their swimwear, and so on. Thin models are definitely in. And again, take a look at more of these pictures. I mean, you rarely, if ever, see a model who isn't rail thin. And it's portrayed also in a variety of different ways in movies, for example, in video games. Young people, you know, are watching these all the time. If you look at children's videos, for example, this is what you see in terms of what bodies look like. And certainly in the case of modeling, as I noted before, this very, very thin appearance. So thin is in our culture, and I think worldwide this is true. Many of you may be familiar with a movie from a few years back, which was called Black Swan, which Natalie Portman portrayed a ballerina who was battling an eating disorder and the emotional and physical consequences this had upon her. If you haven't seen this movie, do see it, because I think it'll help you to understand this eating disorder. Well, some of you may be familiar with the Karen Carpenter story. This was a wonderful singer back in the 70s, the 60s and 70s, by the name of Karen Carpenter, very prolific in terms of being one of the greatest singer-songwriters ever. And she passed away from this disorder called anorexia. And indeed, if you take a look at her, this is her, the year that she passed away, weighing just, you know, 83 pounds. This was her in her prime. This is when she passed away. And she passed away from heart failure at the age of 32. And it's really at this time that anorexia came out of the closet. And by that I mean it was always there, always present, but scientific community wasn't really interested in studying it all that much. And it was not until she passed away where this eating disorder really came to the forefront, and scientists became incredibly interested in studying it. So when we take a look at research on anorexia nervosa, it's an eating disorder that is essentially the unwillingness to eat as much as you need. And it afflicts women a lot more so than men by an order of about 10 to 1. And the age group, which is particularly vulnerable, is about 15 to 24 years of age. There seems to be a genetic predisposition. If you have a sibling with anorexia, then you have a 10 to 20 times, you're more likely to become anorexic yourself. And there's a higher concordance rate in the case of identical twins. When one member of an identical twin pair has been diagnosed with anorexia, it's very likely that the other one will be too. This is in comparison to fraternal twins. So again, genetic predisposition. It's associated with the fear of becoming fat. It's not really this disinterest in food. What we know is that these are individuals who show distortion in terms of their body image. They look at themselves in a mirror and they think that they are grossly overweight. There is recent research that indicates that two neurotransmitters, dopamine and serotonin, may be involved, but in many ways this research is kind of unclear. Don't really know whether or not this is what prompts the disorder or whether or not it's something that results from the disorder itself and the continuation of it. Anorexics suffer from what's called anhedonia. And anhedonia, simply put, is the inability to experience pleasure in your life. This is the highest mortality rate of all psychiatric disorders. For every 100 individuals that are diagnosed with this disorder, 6% will die from it. So again, that's an incredibly high mortality rate. This is an interesting study, one of my favorites in psychology, that was done in the Fiji Islands back in the late 1980s and early 1990s. This island nation, it's a beautiful nation, and here's some islanders that you see here. The incidence of anorexia prior to 1990 was near zero. But then something very interesting happened. Television programming like 90210 and Melrose Place that showed these idealized bodies like what you see here, these female figures that you see here. These television programs became available by way of satellite to island nations like Fiji, and anorexia nervosa dramatically increased. So again, we have to recognize, that this is a disorder that's very much related to this, cultural kinds of factors. So again, we have to be, aware of this as being an important factor. This area of the brain, the hypothalamus that you see here, is, and this is where the brain slice is taken that you see right here. This is about where it's taken. Okay, in the case, this is a rat brain. Okay. If we eliminate smell and taste and other sensory information from the stomach, organisms are still capable of regulating their eating behavior just perfectly. And there seems to be two areas that are involved in this. The ventromedial hypothalamus, here and here. Again, this are the two hemispheres of the brain. Here's one, here's the other one. So the what we call the ventromedial nucleus of the hypothalamus, and again, it's here and here. And the lateral hypothalamus that you see here and here, very much involved in the regulation of this behavior. So when we take a look at some initial clinical observations, patients with tumors or damage to the hypothalamus became hyperphagic. They overate, and they became obese. And indeed, when they did post-mortem examinations on these individuals, you could clearly see the tumors in the area of the hypothalamus. So that really suggested some very important animal experiments that were done to locate, to precisely locate the areas of the brain that were involved in our eating behavior. And indeed, research that was done at the University of Pennsylvania by Alan Epstein as a consequence of these initial clinical observations became very important in terms of documenting this. So the circuitry for feeding behavior, once again, that you see here, lateral hypothalamus here, and the ventromedial hypothalamus that you see here, seems to be the pathways that are involved in this aspect of motivation seem to reside in the hypothalamus. The lateral hypothalamus seems to be that area that's involved in initiating or stimulating our eating behavior. Some refer to it as our feeding center. The ventromedial hypothalamus, on the other hand, that is involved ordinarily in suppressing our eating behavior, so is referred to as our satiety center. So it's these two parts of the brain then that seem to be acting in different ways in terms of the regulation of our eating behavior. Take a look at Alan Epstein. Again, he was a psychologist at the University of Pennsylvania. His very famous studies on stimulating the brains of experimental animals, or actually removing parts of the brain. These studies become instrumental in terms of nailing down this very important factor, very important brain area. So, for example, one of the things that he does is he takes an animal that's just recently had its fill, it has eaten for a period of time and is now stopped, and the animal's satiated. And what he had done is to place an electrode down into the lateral hypothalamus. And again, here's the location of our hypothalamus that we see right here. A satiated animal, even though it's for all practical purposes, it's not hungry, when he stimulates the lateral hypothalamus, the animal starts to eat. Likewise, if he stimulates the ventromedial nucleus of the hypothalamus, and he does so in an animal that's been deprived of food and should want to eat, they quickly stop eating. Okay, so this again indicates that one area of the brain is involved in stimulating eating, and another part of the brain is involved ordinarily in the suppression of our eating behavior. He also does experiments when he actually removes the lateral hypothalamus, and what he finds is the animals become what we call aphagic, that is, they don't eat, and adipsic, meaning they don't drink. You have to force feed them for the first few days after the surgery before they start to eat on their own again. And again, they become aphagic. They start to eat a little bit, but typically nowhere near what they were eating in the past. If you lesion the ventromedial nucleus, what typically happens is, again, the animal becomes hyperphagic and obese. So these are really classic experiments that have been done, which have documented these two areas of the brain being incredibly important in terms of regulating our eating behavior. Here's an interesting figure that you see here. This shows on the left the body weight, which is on the top, and the amount of daily food intake, that's on the bottom here, in animals that have had their ventromedial nucleus of the hypothalamus lesioned. And here is a normal rat that you see here. Here is a rat that has this area of the brain, the ventromedial nucleus, lesioned, and you can see dramatic differences between the two, where the animal that's been lesioned, and again, we'll take a look at its body weight here in the purple, just goes up and up and up and up. I mean, they become eating machines. You know, a 200-gram rat by the end of a 40-day period of time, you know, having received this lesion to the brain, they double their body weight. And you can see this control animal right here. They show relatively steady body weight. And again, this is reflected in the amount of food that they eat. They're eating many more small meals in comparison to that of an adult that has not received that ventral medial nucleus lesion. So again, these are very interesting studies, which once again indicate that those parts of the brain in the hypothalamus are very important in terms of regulating our eating behavior. When you take a look at the characteristics of obese and ventral medial nucleus lesioned animals, you see some very interesting differences. And again, I want you to recall the Schacter experiment with obese animals. They overeat, but only if their food is very palatable. They overeat only if the texture of the food is just right. You know, if it's the texture isn't just right or the temperature isn't just right, they won't overeat. They overeat. They won't work very hard to obtain food. They won't tolerate pain in order to obtain food. If you ask them to cross over an alleyway, for example, in which shock has been administered to the floor, they won't do that. If they have to press a bar many times in order to obtain food, they won't do it. So again, some very interesting similarities between Schachter's work with humans and the work that has been done with lower animals with these animal models have proven to be very important in terms of helping us to understand this this aspect of motivated behavior. There's a short-term system and a long-term system in terms of, you know, the hypothalamus and its involvement in our eating behavior. Short-term system tells you when to start and stop a meal. Long-term system is saying, you know, that the system is involved in maintaining a stable body weight. So we need to know a little bit about those two systems. The short-term factors, certainly blood sugar levels, insulin, glucose, what are called glucoreceptors in the brain. It's the passage of glucose, you know, through those receptors in the brain that's really crucial in terms of these short-term kinds of factors that are involved in regulating our eating behavior. The stomach fullness certainly is another key factor in this short-term regulation. When your stomach is full, the ventral medial nucleus neurons are activated. When your stomach is empty, it's the lateral hypothalamus neurons that are activated. If you take a look also at some wonderful research that's been done on what we call thermosensitive neurons in the hypothalamus, when you know, when the hypothalamus is warmed, then you tend to eat less. When the hypothalamus is cooled, and you can do this by way of these thermosensitive electrodes that are placed into the brain. When your hypothalamus is cooled, your food intake increases. So it makes sense. I mean, when you're in a warm environment, you're not expending calories as much. You're more sedentary. When you're in a cold environment, oftentimes, you know, you are expending more energy. So your food intake is going to increase. So again, we have these short-term factors that are involved. We also have a long-term controlling system, central factors. When there's damage to the ventral medial nucleus, we know that an animal is going to overeat. But if you restrict them in terms of the amount of eating behavior that they engage in, then and they come back down to a normal body weight. But then when you allow them to eat as much as they want again, they just go right back up and become overweight. So it's as if there's a set point in the brain that has been set at a higher level. Damage to the lateral hypothalamus, animals refuse to eat. They have to be force-fed. They maintain their body weight at a lower level. But it appears as though this long-term brain set point has been set lower. So it's almost like a thermostat that's changing. So these are, long-term kinds of factors. Another recently discovered long-term factor that has become increasingly important is what is happening in adipose cells. And adipose cells are the principal difference that you see between a person who's thin and a person who's overweight is in terms of adipose cells, the size of adipose cells. Fat is stored in those adipose cells, and it's responsible for this long-term control of food intake. When you are in a period of fasting, for example, those fat cells release a lot of fatty acid and glycerol, and that glycerol is converted into glucose. Remember, glucose is that fundamental energy source for our brain, for our body. So indeed, those adipose cells seem to be incredibly important peripheral factors. What we also know is that this hormone, which is called leptin, which resides in our adipose cells, again, that's where it's produced. That signals the brain to increase or decrease eating. So if you're in a period then of low levels of leptin, you have increased feelings of hunger, and that stimulates our eating behavior. Some wonderful experiments have been done with animals that are leptin deficient, like this animal that you see here. These are genetically programmed mice that have extremely low levels of leptin. And what happens is these animals become grossly obese compared to animals that do not have that deficiency. So as we learn more about leptin, we're realizing that it's a hormone that's very important in terms of our eating behavior. Again, with that being said, we've looked at a variety of different environmental and biological factors that are involved in our eating behavior. And we'll continue on in our next segment, our next experiment, our next lecture to explore other aspects of motivated behavior.